Hi everyone, it's Mr. Sinti, and today I have the pleasure and privilege to be discussing with you how CRISPR-Cas9 gene editing works, its basic fundamental mechanisms. And so I wanted to begin that conversation with uh, the fact that CRISPR-Cas9, if you have no familiarity with it, is basically an adaptive immune system that was discovered in all places. It was discovered in prokaryotes, both in bacteria and archaea. And so they were able to remember previous viral infections and therefore be resistant to those. And so a true adaptive immune system. Now, I want to remind you that we have an adaptive immune system, far be it more complicated. But I wanted to actually begin this conversation about how CRISPR works today by actually discussing our adaptive immune system, because I think there's some parallel between them, and I think you can appreciate the importance of it. And so the example that I wanted to begin with was a conversation about the vaccine of COVID-19. You might be familiar with this, but I just want to run it by you. The vaccine itself is a nanoparticle lipid bilayer of messenger RNA from the COVID-19 virus. And so what gene sequence is located on that messenger RNA is the gene that produces the protein's spike protein, which allows it to enter our cells and cause infection. But it's also the characteristic that gives the COVID-19 its corona, if you will, and so thus the name. And so what happens is when you receive the vaccine, it's injected into our deltoid, our muscle cells, and then the nanoparticle uh, gets incorporated. It fuses with our muscle cells and it gets integrated. And the messenger RNA is then translated into all things virus spike protein. How about that? So our own muscle cells are producing viral spike proteins on the outside of the cells. And so I'm going to get a little like humorous with this. And so here's our muscle cell. And so what happens is that the muscle cell will be showing the spike protein like this. And so our immune system involves a variety of cells. And so one of the cells, a white blood cell called a neutrophil, is a macrophage, a big eater, and it goes around looking for instances like this of invasion. And so when the neutrophil encounters such spike proteins, it will engulf the cell. And what will happen is it'll take those spike proteins from the cell and actually present it on its own surface like this. And it's what researchers called an antigen presenting cell, an antigen presenting cell. And so this helps to sort of spread the word that there's trouble in the house. So these antigen presenting cells will be cruising along in the bloodstream with the hope that they bump into an ally of theirs, which is another type of white blood cell called the lymphocyte. Now, this is brief, but there's two kinds of lymphocytes. There's T cells and B cells. The first cell that it's likely to bump into is a T cell, but not necessarily. But if it bumps into a T cell, like this, a T helper cell, and the antigen presenting cell, which is a white blood cell that's presenting the spike protein, it will activate the T cell. And when this T cell is activated, it'll start secreting a chemical message called a lymphokine. And lymphokines travel through the blood and they try to communicate the presence of trouble to their friend, the B cell. So the B lymphocyte is the crucial player in our immune response, because the B cell, let me come over here. So here's a B lymphocyte. The B lymphocyte is capable of secreting proteins called antibodies. They're these Y-shaped proteins like this that will circulate in the bloodstream. And so the whole point of the vaccine is to get these antibodies circulating and to get the number of B cells that are circulating them to increase. And so there's a clonal increase and a proliferation of these cells. And so therefore, our immune system, here's the punchline, is primed because it's been tipped off to the presence of the real COVID-19 virus. So if the COVID-19 virus were to come into your body, we would have a very fast and efficient way of responding to it so we don't become ill. So these antibodies would latch on top of the virus and render it inoperable. And so there you can see here. So if this is the real virus, we get sick because we have antibodies and B cells protecting us. So the whole point is, being that it's adaptive, is that we've been primed by the vaccine to respond to future infections. And so this is what's going down with bacteria in CRISPR-Cas9. So as I was mentioning, it's the bacteria, and here's a picture of bacteria growing on a petri dish. It's the bacterial's response to a viral infection. That's what the CRISPR-Cas9 system is really all about. But what's beyond fascinating is that scientists, once that was discovered, are now using creative applications of CRISPR technology to be able to use it in other ways to genetically engineer and alter and manipulate any cell type. More about that in a future video as well. And so to explain what CRISPR does and how it works, first off, CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. Hopefully that will be explained. So it's a type of immune system discovered in bacteria, as I mentioned. These are bacteria shown under the light microscope. What can happen is that bacteria can be infected with phages or viruses, as you can see here in green, and they're terrible. But basically what would happen is the virus will inject its DNA into the cell, and it'll get the cell to read it and start making more viruses inside of it. And so by making more viruses inside of it, it turns the cell into a piñata, and it busts open and it infects other cells. And so what the hope is that once the virus injects its DNA into the host, let me do that again. So once it injects it into the host bacteria, the idea would be, if the bacteria can take a piece of that viral DNA and cut it, and then, you ready for this, incorporate it into its own DNA. So DNA from a prokaryote is usually in one big circle like this. And so if you can incorporate this viral DNA inside, you will then be prepared for a future virus infection. And so this is basically a very simplistic version of how CRISPR works, but I thought I'd start off by mentioning this, that it's using a portion of the virus DNA itself to prime future generations of bacteria and be ready for its presence. Okay, so hopefully that was clear. Let me go on here. So CRISPR are sequences in the genome of a bacteria. So this is the bacterial genome shown below here, that act as a genomic record of previous viral attacks. And so check it out. If one time it was an orange virus, that would be incorporated, or a blue one, or a red one, or a green one. And so what was discovered in the late 1980s in bacteria, although that it wasn't understood to be an adaptive immune system, is they did notice that there were sequences, shown here in black, of repeated sequences over and over and over again that were interspersed by spacers of non-repeating DNA. And as it turns out, that the non-repeating DNA that interspersed was that of phage DNA that had been incorporated. They also discovered Cas genes, which are CRISPR-associated genes, which produce a protein called Cas9, which is a nuclease that actually will cut the DNA of the virus that's trying to infect the bacteria. And so, along with CRISPR, as I was mentioning, Cas proteins are endonucleases that will cut DNA. And so what happens is the bacteria will use the sequences to recognize future invading viruses, so like an actual immune system. And so let me start off officially by explaining what's happening here. So prokaryotic genomes contain short palindromic DNA sequences that are repeated many times over and over. And so what you have here in dark are, let me just highlight it here and I'll go like this. So there's these short palindromic sequences that are repeating here and here and here and here, and they're interspersed by spacer sequences. Okay, so these sequences right here called spacer are sequences of phage DNA that have been incorporated. And so the palindromic, the word palindrome is a sequence of letters that are spelled left to right, and they spell the same thing, like never, odd, or even, or the word kayak, as it turns out. And so you can see an example of that here, palindromic sequences. And so these repeated sequences were initially called clustered, regularly interspersed short palindromic repeats, or CRISPR. And so that's where the name CRISPR comes from, that these palindromic repeats were interspersed by spacer regions. And so each palindromic repeat is followed by a short segment of spacer DNA. And that, as I mentioned before, the spacer DNA is what actually matches the sequences found in bacteriophages. And so those were viruses that were known to infect bacteria. And so what you have here is spacer DNA, then you have palindromic repeat, spacer DNA, palindromic repeat, spacer DNA. And so this became known as the CRISPR array. And so these palindromic repeats, the sequence of nucleotides in both the same direction, were the result of RNA forming a multiple hairpin loop structure. And as it turns out, that when it's palindromic, it tends to fold back on itself like that. And spoiler alert, it was these repeating sequences of DNA and then transcribed into RNA. It's this so-called tracer RNA that helps to associate or connect the RNA with the Cas9 protein, as it turned out. So what's interesting is the spacer DNA is not identical. And so when that was sequenced, the spacer DNA, and then it was run through bioinformatics, and it was identified as being similar in sequence to phage DNA, it became clear that what we were dealing with is sort of a memory of past infections were being integrated into the bacterial chromosome itself. And so when researchers began to sequence the spacer DNA, as I was saying back in the 80s and 90s, it puzzled scientists because the database wasn't as complete as it is now, and it gets better each time new sequences are entered. But around 2007, researchers started to put together these curious facts, and they demonstrated that CRISPR and the Cas genes work together to form an immune system against phages from future infections. And so, as it turns out, the spacer DNA matches up perfectly with viral DNA, especially bacteriophages. Okay, and so the insertion, let's see if I can get that cursor back, I guess not. So the insertion of multiple spacers in a bacterial chromosome is correlated with increased resistance. So the more phage that infect bacteria, their descendants will have a greater resistance against any of those viruses that previously infected them. And so, let's see. Trying to erase, but not having an easy time of it. But I guess I'll go with that. So they identified a number of genes associated with CRISPR, as I was mentioning. So these CRISPR-associated genes are the ones that make the Cas proteins. And so those are upstream from the CRISPR array. And so these genes over here are the ones that are going to be transcribed into the endonuclease Cas9, which is going to be cutting the viral DNA upon a future infection. And so let me see if I can, there we go, better. So the Cas protein unwinds and it cuts DNA. So the basic principle of CRISPR and how it works at its very core is that it's basically a protein-based enzyme called Cas9 that is an endonuclease that cuts DNA of viruses that are trying to infect bacteria. And it's like, well, how can they do that? Well, the Cas is a DNA cutting enzyme, but its specificity in terms of the DNA sequence is that it's recognized by a sequence called guide RNA. As it turns out that the insertion, if you will, so here you have a spacer. The insertion of a spacer DNA inside the genome of the bacteria will result in messenger RNA. And that RNA will then guide the endonuclease. As you can see, here's the RNA right there. It will guide the Cas9 enzyme to the virus that's infecting it. More about that. Okay, let's see. Okay, there. So when a virus injects its DNA into the bacteria, what will happen is there's other Cas proteins involved, something called Cas1, Cas2, and what those enzymes can do is basically cut up the invader DNA sequence, right in here shown in green. And so once that viral DNA is cut, it can become a new spacer, and it's added to the growing CRISPR array. As you can see here, these color blue, purple, and orange are previous infections. And so now this green is going to be added to the bacterial chromosome. And so the invader is then added to the clustered, regularly interspersed short palindromic repeats as a way of serving as a memory for what that virus looked like that was trying to hurt the bacteria. And so the CRISPR array is a short stretch of DNA, as I mentioned before, that's found in bacteria, and it's composed of alternating sequences of the targeted specific spacer sequences and then these repeat sequences. And so the segment of viral DNA that is being cut is known as a protospacer. And so what will happen is the Cas1 and 2 will cut the protospacer DNA, and then it will become inserted into the CRISPR array. And it'll add to the memory that the bacteria will have of the viral infection. So if the phage returns, so this is the point, if the phage returns, the bacteria is sort of armed to defend itself against that. It's like, oh, look at this. I've seen green phage before. And so what it will do is it will produce, let me go over here, make that a little smaller. It'll produce right here a piece of RNA that will target the viral sequence like this. And then it will also produce an RNA that will form a loop like this hairpin loop called tracer DNA, which will then help it to become connected to the Cas9 endonuclease. And then when all of this is in place, the Cas9 enzyme will cut the foreign DNA and therefore render it inoperable. So that's pretty cool. And so let me go on here. Okay, so this defense starts with transcribing from the DNA, transcribing the long CRISPR into RNA, known as CRISPR RNA. And from the repeats and spacers of the CRISPR array, you get what's called the pre-CRISPR RNA. And then, as I'll just mentioned, that's transcribed. And then those are all cut up into individual units, which will then detect the viral DNA of a future infection. And so the guiding region, part of the guide RNA, the CRISPR RNA, that is typically around 20 nucleotides in length, 20 nucleotide base pairs, or just nucleotides, I should say pairs, that will be complementary and will pair up with the target DNA sequence, and that'll be where the Cas9 DNA protein cuts. So the guiding region of RNA contains a sequence that matches the invading virus. And what will happen is this other RNA, as I mentioned from the repeat unit, is called the transacting or tracer RNA. It comes along and links up with the CRISPR RNA through base pairing, like this. And it becomes a complex which then binds with the Cas9 protein. And then that becomes an effector against foreign invasion. And what will happen is the Cas will then take those dual RNA sequences and trim them and make it into a more manageable complex. And so this CRISPR-Cas9 will search out for, and there's many different ones, will search out and try to defend itself against viral infection. If the sequence of the CRISPR RNA matches the sequence of the invading virus, then it will destroy the phage by allowing it to cut the DNA, and therefore if it cuts the DNA of the virus, the bacteria will survive. So it's an adaptive immune system which prevents it from being destroyed by viruses. And so Cas9 is a nuclease, which is a type of enzyme, as I've been describing, that cuts DNA. And so when matching the sequence, the matching sequence known as the guide RNA finds a region in the viral DNA, it'll complement and pair up with it, and then the Cas9 will actually make the cut, disabling the virus. And so as you can see here, it would seek out the sequence of the virus and then cut it. But if you're following this, you might be saying, but wait a minute, if the Cas9 protein can recognize certain specific sequences, what would prevent it from cutting its own CRISPR array as well as cutting the new viral DNA? And like, how is it able to distinguish itself from the enemy, if you will? And so this is where something called the PAM comes in. The PAM is a region of nucleotides in the virus DNA that helps Cas9 identify it as being the true enemy and associate with it and cut it. And so PAM stands for protospacer adjacent motif. So it can vary in length, but mostly it's a specific sequence of nucleotides, around 3 base pairs, that follows the protospacer sequence found in the viral genome. So if this is the viral genome, it's a sequence of 3 bases that follow at the very end of it, and it's known as the protospace adjacent, right next to it, motif, or PAM. And so Cas9, it won't cut the target DNA unless the PAM sequence is present on the protospacer DNA, the target DNA. And so it binds to that PAM region, and therefore it's able to make the cut. And so PAM is very important. Therefore, the Cas9 gene would not be cutting up its own DNA. And so the PAM sequence must be present for the Cas9 protein to know that it's okay to latch on to this foreign DNA and cut it. And so how does this keep the bacteria from hurting itself? Well, as I mentioned, it won't cut its own DNA, because the key is that the spacer sequences don't have the PAM. And so as you can see here, they intentionally don't have the PAM. And so the sequence GG is an important one in identifying a typical PAM motif. It's usually NGG, N meaning that it could be any of the nitrogen bases, AGG, TGG, CGG, GGG. And so the CRISPR DNA doesn't contain that. And so the protospacer can be, or the PAM in the protospacer can be any base followed by GG. So Cas9, to review, recognizes the PAM sequence of the 5' NGG, where N could be any nucleotide. It recognizes it, not to get too complicated about it, but the Cas9 protein has a particular sequence of amino acids, which then associates with the PAM, holds it in place, and then the cut is made, based here on what the recognition sequence being complementary to the target sequence. And then the scaffolding area right here, the tracer RNA, is what helps it associate with the Cas9 protein. So the protospacer adjacent motif, or PAM, has to be present. And so if it's not present in the DNA, then Cas won't do its thing and cut the DNA. So it has to be present. So here's the 20 nucleotide target binding. There's the correct PAM. Here's the tracer DNA, which helps it to incorporate with the Cas9. And so when Cas9 binds to the PAM, it separates the DNA adjacent by binding with it, and then it will open it up, and then if it's complementary to the sequence, then Cas9 will cut the DNA. And so the scaffolding region, again, I mentioned this before, that is tracer RNA. And in nature, it's a region that forms the hairpin loops, thus the need for palindromic sequences. And it helps to attach to Cas9 and allow it to work properly. Fairly recently, scientists have created something known as Single Guide Rna, and it's an engineered form of this, of what I've been talking about. Instead of having two RNAs, it's a single guide RNA, which works very simply. It's about 100 nitrogen base nucleotides long, and it fuses the two important areas. It fuses sort of the tracer RNA to this guiding region area. And so when you have something like a single guide RNA, what you could do is manufacture this in the lab and actually create any nucleotide guiding region, and it will work because it will bind, provided there's a PAM, but it will bind to the Cas, then it would complementary anneal to any particular sequence you're interested in, and Cas9 will do its thing and cut the DNA. So this single guide RNA has been a game changer, to say the least. It's a single, as I mentioned before, a single unit which can then track down any specific sequence. So it's a chunk of RNA with a structure that allows it to pair with any system that is specific by binding to that 20 complementary base sequence. And so it's rather remarkable. And this is what allows it to be capable of doing some very interesting and profound genetic engineering achievements, both present and in the future, the single guide RNA. And so biotechnology was quick to see the power of this, because you can program this 20 nucleotide sequence of the single guide RNA, and basically it's like a restriction enzyme that is targeted to wherever you want it to be, and then it will cut the genome right where you want it. So you can cut it and add or delete from any genome using a single guided RNA. And so this is really impressive. So Cas9 is, how does it work? It's a RNA guided endonuclease that creates double-stranded breaks in viral DNA. It's activated once the CRISPR RNA and the tracer bind together, or if you're using a single guide RNA, it will recognize the DNA and bind to it, provided that there's a PAM site. And this is a cool image here of a 3D printed Cas protein showing its ability to adhere to DNA. And so here's a couple of like cartoonish models to sort of walk you through the steps of what I've been trying to describe. Is that the first thing that takes place is that the single guided RNA will bind to Cas9 protein. So there it is. And then when that single guided RNA is attached to the Cas9, it'll start to search foreign DNA, and it'll start to look for PAM sites, and it'll proofread it and try to also find where the 20 nitrogen bases are complementary. And if Cas9 does recognize the PAM sequence, then like maybe it'll make a cut if the sequence is complementary, provided that it finds the NGG. And so as it turns out, there could be more than one PAM site present, but if the sequence isn't complementary, then no cut will be made. And so here's an example of, oh, look at this. Here's a PAM sequence that we're going to bind to. And look, there's an absolute perfect complementary sequence. And so at this point, the nuclease Cas9 can engage and cause double-stranded cuts in the DNA. And as a result, cut will break the DNA up. And so it will prevent the bacteria from being infected. So to review this, researchers can synthesize the single guide RNA, a sequence of about 20 nucleotides that matches a particular sequence of a cell's DNA that they want to target. And then when the guide RNA is added, the Cas9 will connect to that and basically look for the PAM, or protospace adjacent motif, and then in some way it'll bind to that, and then if the location is correct, it will complementary bind, and then it will cut. So step 3 is where it will actually make the cut and cleave the DNA upstream of the PAM. And so if the DNA RNA pairing triggers Cas9 to change its three-dimensional shape, it allows it to make a cleave. And now the fourth part of this, and I hadn't mentioned this previously, is that cells don't particularly, when we're talking about we're not talking about bacterial defense anymore. We're talking about if this was used to cut DNA. Cells generally don't like their DNA being cut. And so what a cell will do in response to a break, a double-stranded break, is that basically it'll try to repair it, because it's something that happens on occasion. There's breaks sometimes that it will occur, and so you need a repair mechanism. And so the repair process is naturally error-prone, especially in bacteria. When they have double-stranded breaks, they are capable of repairing it, but often the repair to the broken area is random. And if it's random, it probably will result in something that won't be functional. And so CRISPR provides the cut, but there's other enzymes involved in the repair process. And so they don't, as I was saying, they don't like to have double-stranded breaks in their DNA. And so a double-stranded break in a bacterial genome for too long is usually lethal. And so when there's a break, repair enzymes come in and they try to put in some nucleotides. And so I want to basically describe, because this is important process in terms of repair mechanism. There's two basic repair systems at play here. One of them is something called the non-homologous end joining versus homology-directed repair. So let me talk about the non-homologous end joining first. And so when there's damage done to a cell, the non-homologous one, which is more frequently used, as I was mentioning, it doesn't have a template to use to repair the break perfectly. And so basically it's just joining in any nucleotides together. And so it's error prone, which introduces mutations in the target sequence. And so those mutations could can introduce some kind of problem to the gene to make it dysfunctional. It could be an insertion or a deletion which throws off the reading frame. It could be a premature stop codon, but it's usually a problem. And certainly if it's able to survive that, repeated cycles of cleavage and damage would certainly result in trouble or a mutated sequence. And so this type of mutation, as I was saying, is random. And so what can result from this is that it likely inactivates a gene. And so what scientists realized when they were discovering this, they're like, oh my goodness, like this actually could be harnessed for good. Like if we can target a particular sequence in a cell that we know that we want to disable or knock out, we could basically target a single guide RNA to that region, have Cas9 cut it, and when it repairs, it'll make mistakes and it'll basically knock out that gene and neutralize it. And so some ways we could use the fact that the gene doesn't work anymore to figure out phenotypically what the gene was even doing in the first place, because sometimes we don't know. So basically rather simple things like knocking out genes by making mutations at specific positions in DNA is rather useful, as it turns out. But this homology-directed repair, HDR, is even cooler still, because if we can direct single-guided RNA Cas9 to cut a particular sequence that we're familiar with it, we could also insert into the cell a piece of DNA that will be basically familiar on both sides of it. And so when the cell is repairing, it will use these homology arms, if you will, to integrate this template DNA into the genome, and it will therefore replace the break with our own new sequence. So a really cool way of editing and introducing a sequence that we care about. And so scientists can manipulate this repair system by introducing the cell a template along with the guide RNA complex that sort of tricks the cell's machinery to integrate this using this repair template to break and then fix it with homology-directed repair. And so by designing different templates, scientists can change the target DNA of any cell by creating a mutation or correcting a mutation or causing a break and then introducing a new piece of DNA. So rather powerful. So made up of DNA cutting enzymes, Cas9, and programmable RNA molecules such as the single guide RNA, and CRISPR-Cas9 can precisely target nearly any gene. And so the idea is, how do you get this into cells that don't normally possess it? And so if you're working with bacterial cells, it's rather simple. You can actually insert using plasmids as a vector in transformation. By heat shock, you can introduce plasmids that contain repair enzymes. You can introduce the Cas sequence, the Cas genes. You can insert single guide RNA. You can even insert homology-directed repair template sequences to alter bacterial sequences. Now, when it comes to eukaryotic cells, you would need to use electroporation, but basically you can introduce any of these single guide RNAs or Cas9 sequences into any cells in order to genetically engineer them. So think of Cas9 with the help of single guided RNA as basically molecular scissors with a programmable GPS that can go to any particular target sequence. It's rather impressive, to say the least. And so what I wanted to do now is show you, if I can get this right over here, I wanted to show you this one segment of a video that kind of does a wonderful job explaining this.
Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9,
which is the nuclease,
and an RNA molecule known as the guide RNA.
So the guide RNA, as I was mentioning before, is kind of a hybrid that was engineered synthetically of the tracer and the CRISPR area that was capable of finding the target sequence. So all you have to do is change the sequence that recognizes the template, and you basically can take this whole system and direct it anywhere you want.
Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA.
Usually 20 nucleotides in
Once
length.
it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene.
Or knocks it out.
This makes CRISPR a great tool for knocking out specific genes.
Okay, and so that's surprisingly a brief look of how CRISPR operates and how it works. Hopefully that was informative and you enjoyed it, and thank you for watching.